Hi, and thanks for joining us today. We're going to talk about gene therapy vector development. There are a lot of different aspects in the gene therapy research field, and a lot of it starts with having the right vector to deliver your genetic material to whatever target you're interested in the cell. So the process of optimizing your vector can be really crucial for the long-term and ultimate clinical success of whatever gene therapeutic project you are trying to develop. So with all that in mind, what I wanted to do today was take some time to talk about what techniques exist in the field to assess the quality and the genetic fill of your adeno-associated virus, or AAV, and then go into some of the solutions that NanoTemper offers for this and why really producing high quality data is so crucial to ensuring your success in the market. So I'm Dr. Stephanie Call, and I have been working on the Ponto product team for quite some time. And so I have a lot of personal experience with how the gene therapy field is going, where it's headed, and what we can do in order to improve your research outcomes. Okay, so let's get started and talk about what considerations gene therapy researchers have when it comes to vector development. The first is the serotype of their vector. What vector is going to give you the desired tropism, and was it expressed and purified as expected? The next is formulation. So in this idea, we're thinking about what conditions keep the vector stable and allow it to behave well over time for both experimental setup and for long-term storage. Next, we have the question of fill. Does your vector contain the genetic payload, and how much of it does it contain? And last, we're going to address concerns about limit of detection. AAV samples are incredibly precious, and we want to use as little sample as possible in order to make good decisions about how to use the AAVs going forward. I want to start by briefly addressing what serotyping is and how it has an impact on the stability of an adeno-associated virus, or AAV. Again, today we're largely going to be focused on AAVs. There are obviously other gene therapeutic delivery systems, but most of our focus here is on AAV. There are 8 commonly used serotypes of them, and they are comprised of 3 viral particle proteins. The different sequences of these proteins is going to give them different surface properties and tropisms, and it is the 3rd viral particle protein that is largely what determines the overall stability of the capsid. And the methods that are at gene therapy researchers' disposal for optimizing stability are buffer formulation, doing site mutations to slightly modulate its behavior, and then doing further covalent structural modifications after the purification process. I want to get into talking about the different methods that are available for profiling gene therapy vectors and some of the benefits and drawbacks of each. The first is analytical ultracentrifugation. This follows the intrinsic fluorescence of a protein, so there's no additional dyes or anything needed for it. And it is a well-established method that allows you to assess how homogeneous your preparation is, so ensuring that there's no aggregation or anything present. And for measuring the hydrodynamic radius of your particles, so how large is your associated particle, and then also whether your vector contains the genetic material and to what extent it contains it. Is it 100% full, 50% full, or 0% full, completely empty? Now, some drawbacks of analytical ultracentrifugation is that it is not considered a GMP compatible technique. There are a lot of concerns about reproducibility and accuracy when we think about experimental design for it. Additionally, it requires a long time to run a single AUC experiment, and it requires quite a lot of sample in order to get that good separation that you need in order to assess these parameters that you want to know. Next up, we have extrinsic differential scanning fluorimetry. This is commonly done with Cipro orange dye, which binds to the hydrophobic patches of a protein and therefore can be used to measure the melting temperature. So as a protein unfolds, it binds to the newly exposed hydrophobic patches and gives a signal. This allows you to optimize your formulations to attempt to increase that melting temperature. A more thermostable protein is expected to be more thermostable over time, and the same applies to a fully assembled AAV. One of the great advantages of this is that it's cheap and easy to use. Almost every lab has a fluorimeter that you can use to do this kind of measurement. However, some drawbacks is that because of the way the dye works, it can only see a single transition. And additionally, when you're thinking about excipients or stabilizers in your buffers, not all of those additives are going to be compatible with this dye because it might trigger fluorescence prior to the onset of the experiment. The next two techniques I'd like to address are ones that we at NanoTemper offer, and they're dynamic light scattering and nano-differential scanning fluorimetry. So nano-DSF is similar to the extrinsic fluorimetry that I just mentioned, but it uses the intrinsic fluorescence of your protein. The absorbance behavior of your protein is going to change based on whether it is folded in or unfolded and exposed to solution. And what we can do is monitor very closely the two different wavelengths necessary to determine the unfolding of your protein without the use of extrinsic dyes. Next, we have dynamic light scattering. This is a fairly complicated technique, and I don't want to spend too much time today getting into the weeds of the technical there. But the main take home to realize is the parameters that DLS can give you. And there are two main components. One is the hydrodynamic radius can be determined from DLS. So that's the size of your VLP or AAV or whatever you might be associating, as well as the dispersity. So if there are higher order oligomers, or if the association of your vector is not very good, then you might see this peak broadening. And that is indicated by a PDI or polydispersity index value. A PDI that is close to 0 is a very nice, tightly folded single species. And a PDI that starts to get closer and closer to 1 is basically a bad sample. It's got a lot of mixture in it. It's not well folded. It's not well associated. So you want to avoid polydispersity during your development process. Like I said, the Prometheus Ponto offers both the DLS and the nano-DSF technology. And today we're going to look at specifically how the Prometheus has enabled researchers to improve their vector development through serotyping, monitoring the loading of your vector, as well as evaluating how expression and formulation have an impact on the overall stability of your vector. So let's get into it and look at some real data from the Prometheus and understand why this high quality data is very, very valuable and important when making long-term decisions about your gene therapy projects. The first comes from this paper it's a book chapter that I really like that goes into a really deep dive on how the Prometheus completely expedited the process of evaluating and choosing the optimal formulations for different AAVs. This is a pretty straightforward serotyping example, and the main takeaway here is just that the different vector serotypes have different behaviors. And that's because of subtle sequence differences in those viral particle proteins that comprise them. The first derivative view is useful for seeing where those inflection temperatures take place more specifically, and just creating this baseline kind of fingerprint for your vector is the first step in then optimizing the formulation and behavior of it for long-term use. Moving on, one of the specific examples of why nano-DSF and the intrinsic fluorescence is valuable is this example here. I'd like to draw your attention specifically to this dark green line, which is actually a 50-50 mixture of AAV1 and AAV2. With extrinsic fluorescence, it would be very difficult to see the two separate transitions that each of the AAVs contributes, but by using nano-DSF, we can see both of those transitions in a single experiment and be able to optimize for the stability of each of them going forward in kind of the buffer formulation or other optimization assays. Next, I want to talk about the impact of stress on the quality of your AAV. Here we have AAV2 before and after 5 freeze-thaw cycles. This is from the same work that really demonstrated how the Prometheus can expedite your gene therapy research. And I would just like to draw the attention to the decrease in quality that is not only evidenced by the slight but moderate shift in TM, it's only about 2 degrees here, but the real change in quality can be seen by the change in the initial fluorescence ratio. You can see that we have a higher initial ratio right at the onset of the experiment, and that there's a more gradual unfolding slope leading to the inflection point. And what that means is that this stress-exposed vector in general is just less stable compared to its native prep. We would expect that result, but it's very important for researchers to be able to evaluate how much stress exposure is tolerable for their long-term research goals. Additionally, I'd like to address the question of concentration. Anybody who has already worked with gene therapy vectors knows it's pretty difficult to get them terribly concentrated, and there's a fairly relatively narrow range of concentrations that generally AAVs are worked with. So here we have the yellow is actually the highest concentration, and then green, red, and blue, all decreasing in concentration. And what you can see here is that the melting temperature does not change significantly, but there is this broadening of the first derivative peak, which can be more clearly seen by the slope at the inflection point, so how sharp this inflection point is here, versus the sample concentration. And again, as this peak broadens or as the slope gets less severe, that is an indication of decreased stability compared to another sample. However, the counterpoint is as well, this low concentration is 2 times 10 to the 12th vector genome per mil, and it does show that we're still getting the same melting temperature when that measurement is taken. So if you're not specifically trying to find the optimal concentration for storage or assay development, it's still important to consider that you're getting the same melting temperature information even at a fairly low limit of detection. So it's about personally evaluating what is key for you when it comes to looking at these different parameters you need for stability assessment. Next, we have a pretty straightforward example of buffer formulation. This was done by an associate group, and all they did was screen an AAV in 2 different buffers, 1 and 2. And again, serotyping just shows really clearly buffer 2 behaves a lot better compared to the AAV in buffer 1. Sharper transition, less gradual slope. So thinking about scaling this up to a full buffer screen, whether it's 96, 384, the Ponto can really allow you to expedite this process and quickly assess which buffers are giving you the highest quality AAV environment. This is an example from a group that published here, and I love it because it really drives home the importance not just of the assembly, but also the production method. So here there are HEC293 cells compared against SF9 produced AAVs, and then these are both in SF9 cells, but produced by different production methods. So those of you who are working in gene therapy might be receiving your vectors from someone else and not actually even building them yourselves. And it's really crucial to have this quality control when you're thinking about getting these vectors from different sources, not just sources as far as the cores, but also just what expression system are they produced in. The different expression systems have different glycosylation patterns that may ultimately affect your tropism and efficacy for the vector you're concerned with. So having this finely tuned nano-DSF data is really critical for comparing batch to batch efficiency, whether you're getting the expected results for your further down the line experiments. And this is a quick and easy way to make sure that the product that you're getting or the product that you're producing is high enough quality to move forward with. Next, I want to start to get into the data that we have done. For those who don't know, we recently launched this Prometheus Ponto that has DLS. And it was very important to us when working with the gene therapy space to ensure that our instrumentation lined up with the expectations in the field. So we began by comparing work done in our Prometheus Ponto against that done previously published in this paper. These melting temperatures were determined from the extrinsic fluorescence technique, so using the Cypra orange. And then the values in blue represent the Prometheus Ponto. And the main take home here is just that the data that we're seeing from intrinsic DSF lines up very, very well with the literature value. And additionally, a benefit is that the Prometheus Ponto allowed a lower limit of detection and less material usage overall compared to what was previously published in the literature. Zooming in a bit on the empty versus the full capsids, here we have empty versus full for 5 different capsids that did. There's not always a huge change in the inflection temperature between the AAVs based on their fill status. However, there can be a difference. AAV2 shows it quite starkly, and there's actually a significant enough difference for 1 and 6 as well. Again, using the Ponto, we get highly reproducible data, which allows us to have these nice tight error bars that allow us to make very good decisions about if there's a significant difference between the behaviors. Now, thinking about that a step further, it's more important to be able to predict the fullness of your capsid. So what we did to validate this was to actually look at the initial fluorescence at a single wavelength. So here I have the fluorescence at 330 nanometers. And we created basically a calibration curve for each one of these serotypes based on the fill status of their vector. And the 2 main points I want to point out here, one, once again, each serotype is different. Whenever you're doing this kind of work, you always have to consider that every serotype has its own unique properties and behaves differently from a parallel or different serotype. Number 2, we did these experiments in triplicate, and you can see super tight agreement, great linearity between these different experiments. Which really gives you confidence that if you are probing an unknown, once you have created a good baseline plot, it's very easy to quickly determine how full or empty your unknown capsid is based on these plots created here. Just to drive home that point a little bit, we also pushed the limit of detection for this as well. You can see great linear agreement no matter what concentration we're working at. And again, there is subtly different behavior, so you do have to be cognizant of what concentration you're working at. But overall, this linearity agreement is really great and is very useful for those who need something much faster and more straightforward than analytical ultracentrifugation when it comes to assessing the fill level of your vector. Now, lastly, I would like to address why dynamic light scattering in parallel with the nano-DSF is really useful for vector development. Here I have an example of AAV1 in both its empty and full form. And from the intensity distribution from DLS, you can see a nice sharp curve where that vector is formed. There are not too many higher order species. They both give PDIs of about 0.24, which are considered pretty good PDIs, especially for a complicated preparation such as a viral vector. However, anybody who has worked with any of these AAVs knows that AAV2 is notoriously stubborn to work with, very prone to aggregation. And that is very evident from the DLS data here, where we see these higher order aggregation distributions up in the hydrodynamic radius range. So while there is some single AAV present, a lot of the material has been lost to aggregation. And obviously this is going to be very important to evaluate when thinking about delivering your sample to patients. There can't be contaminants or aggregation because that's going to ultimately impact the efficacy of your vector. So DLS is a really quick, easy way to assess how your sample homogeneity is and whether it is optimized for downstream assays. So I want to bring everything back to the top and just kind of summarize really quick. The Ponto allows you to consider many important questions about gene therapy vector development. The first is the serotyping, which is just getting your fingerprint of your AAV and seeing how it's behaving in a given environment. Next, we have the formulation, which is about optimizing for a more thermostable and a lower PDI, so more homogeneous sample. Next, we have the fill. So questions about the genetic payload and how much of it is in the vector are very crucial when we're talking about going forward and delivering the genetic payload to patients. And last, limit of detection. Ponto allows a very low limit of detection compared to other methods in the field. And with the low yields of a lot of these AAV samples, being able to make good decisions and still get quality data, even at the lower limits of detection, is completely crucial in getting good data and being able to make the best decisions about your therapeutics. So with that, I thank you all for paying attention and for joining us today. My colleague Leah is going to get me started with the Q&A, so I'll address as many questions as I can. And thank you guys for sticking around this long.